I was asked to talk about practice-changing research in neuroendocrine tumors, both the, let's say, recent past and present, and give a little bit of thoughts about, patient role in all of this. So let me just start out with a few definitions, and Josh has already very nicely defined some of these things, but we talk about phases in clinical trials. So phase one are early human trials. In other words, the drug looks promising in preclinical, maybe animal studies. And these are typically dose escalation studies, starting at a very low dose and escalating up as tolerable, with the primary purpose of evaluating safety and establishing a safe dose for subsequent research. Phase two trials are primarily designed to assess the efficacy of the drug. Usually it's a single-arm study with endpoints such as response rate, meaning tumor shrinkage, or time to tumor growth, or progression-free survival. Usually these are single-arm studies, but occasionally a large phase two study can be randomized. And then phase three are the large, typically randomized clinical studies comparing the new drug against either a placebo or a standard of care, usually designed to prove that the drug is superior to either of those things. And typically the goal of a phase three study is to get the drug registered and approved. The most common endpoint in neuroendocrine tumors studies, especially phase three studies, is progression-free survival, meaning basically time until tumor growth. Sometimes the endpoint is response rate, meaning the percentage of the patients with significant tumor shrinkage. Overall survival is not typically a response rate because it would require usually thousands of patients in a randomized trial to be able to prove overall survival benefit, and we just don't have these numbers of patients. But it's often a secondary endpoint. And then quality of life, and I think Josh described that well. So what are the major trials of the last decade or so? All of you know these trials to some extent, so I'll just really go through them very briefly. The two somatostatin analogs, octreotide, sandostatin, lanreotide, somatulene, those were both proven to inhibit tumor growth in their respective studies. ProMed compared octreotide versus placebo in metastatic midgut nets and showed significant improvement in time to progression. Clarinet looked at lanreotide, somatulene versus placebo, and also showed significant time to tumor growth. So despite the fact that neither drug causes significant tumor shrinkage, they both inhibit tumor growth and were proven on these two phase three studies. Everolimus, Afinitor, was investigating the RADIANT studies. These were RADIANT 2, 3, and 4 were all phase 3 studies. RADIANT 2 looked at everolimus plus octreotide versus placebo plus octreotide in patients with progressive neuroendocrine tumors and history of carcinoid syndrome, so mostly midgut neuroendocrine tumors. This study, even though the curves look a little bit separated, was technically a negative study. The difference was just short of statistically significant. RADIANT 3 looked at everolimus versus placebo in metastatic pancreatic neuroendocrine tumors and showed a significant improvement in progression-free survival. And then RADIANT 4 looked at patients without history of carcinoid syndrome, non-pancreatic nets, so mostly colorectal, lung, some midgut, and it also showed a statistically significant improvement in progression-free survival. So these were all phase 3 studies of everolimus. There was also a phase 3 study of sunitinib, Sutent versus placebo in metastatic pancreatic nets, and that also showed a statistically significant improvement in progression-free survival, very similar to the RADIANT 3 study. ECOG 2211 was a randomized phase 2 study, so almost as large as a phase 3, but not quite. There were about 140 patients. It compared capecitabine temozolomide versus temozolomide alone, and it showed a significant improvement in progression-free survival with capecitabine temozolomide. The NETTER-1 was a phase 3 study looking at lutetium dotatate versus high-dose octreotide, and it showed a very significant improvement in progression-free survival that was highly statistically significant. And then more recently, there were two Chinese studies looking at surufatinib, one in non-pancreatic nets, the other in metastatic pancreatic nets. These were both positive phase 3 studies showing improvement in progression-free survival, but the drug was not approved in the United States and Europe, based mainly on the fact that the studies took place in China and they wanted to see multinational studies showing this efficacy. There are two studies that actually were just presented at ESMO last week. One was the OCLU random study. That was a randomized phase 2 study looking at lutetium dotatate versus sunitinib in metastatic pancreatic NETs. And it showed a very clear superior progression-free survival with lutetium dotatate versus sunitinib in patients with progressive pancreatic NETs. But it was a relatively small phase 2 study, only 80 patients in total. The SECTOR study was supposed to be a sequencing study looking at everolimus followed by streptozotocin 5-FU chemotherapy compared to streptozotocin 5-FU followed by everolimus in metastatic pancreatic NETs. There were various issues with the trial accrual, as well as I would say with eligibility criteria. Patients did not need to have progressive disease at baseline, and that really, I think, impacted the study. To the point that there really was no difference between the two arms in terms of progression-free survival, although not surprisingly, there was a higher response rate, meaning tumor shrinkage with the chemotherapy regimen, streptozotocin 5-FU. So those are two very recent studies. What about studies in progress? So I want to highlight the studies of alpha-emitting PRRT. I think many of you have heard about alpha emitters. This is a type of peptide receptor radiotherapy with isotopes that emit basically 2 protons and 2 neutrons. So a much heavier particle than the previous isotopes, lutetium and yttrium, which emit beta radiation, which is basically electrons. So it's much higher energy at a much shorter particle range. We are hoping that these drugs result in better outcomes and less toxicity. So there's 2 isotopes being studied. One is actinium-225. That's the RayzeBio study. There's a phase 1 portion, which is taking place now, which will be hopefully followed by a phase 3 portion where the drug is compared to one of several standard of cares in patients with progressive NETs. And then the other study is a single-arm phase 2 study of lead-212 dotamtate, which is another alpha emitter. This has 2 cohorts, one in patients who are naive to PRRT. In other words, they've never received lutetium dotatate or any other PRRT. And then there's going to be a cohort of patients who have received lutetium dotatate. This study is looking primarily at objective response rate, or percentage of patients with significant tumor shrinkage. So 2 studies looking at alpha emitters that are quite exciting. There's also studies looking at lutetium dotatate as an early line of therapy for relatively aggressive grade 2 or 3 NETs. That's NETR2. And then another study looking at lutetium dotatate, which is very similar, also as an earlier line of therapy. That's the COMPOSE study. There's also the COMPETE study, which is looking at lutetium dotatate versus everolimus in patients with progressive neuroendocrine tumors. That's just completed accrual last month. Another large study is the CABINET study looking at cabozantinib versus placebo in patients with progressive pancreatic as well as non-pancreatic neuroendocrine tumors. That's a very large phase 3 study with 2 cohorts that's looking at this tyrosine kinase inhibitor. There's a study for liver-dominant disease. It's called the RETNET study, looking at bland hepatic arterial embolization versus chemoembolization. These 2 embolization modalities have been available for many years, but no one has ever really compared the 2. So this is the first study comparing different types of embolization. And I also want to finally highlight a study of adjuvant capecitabine temozolomide versus placebo in patients with pancreatic neuroendocrine tumors that has been resected. Adjuvant means treating patients who have no evidence of disease on scans, but who are thought to have a high likelihood of microscopic disease after surgery. And the point is to test whether chemotherapy can eradicate that microscopic disease and either prevent recurrences or delay recurrences. So this is really the first adjuvant study that I'm aware of in this field. So Josh has mentioned quality of life, which is typically a secondary endpoint in some of these studies, especially the larger studies. There are several widely used metrics, including EORTC. There's the FACT-G metric. There's a less used neuroendocrine quality of life. Both general questions as to quality of life and overall health. For example, how would you rate your quality of life over the last week? How would you rate your health? What is your ability to perform normal daily activities like going to work or do hobbies? And then specific symptom questions like how would you rate your diarrhea or flushing, among many other symptoms. Patient reported outcomes are similar, but not necessarily graded on a quality of life scale. They might just simply ask how many times a day do you have diarrhea, or how many times a week do you have flushing or abdominal pain or other symptoms. These are most useful in randomized studies where one can be compared to another. Less useful in single-arm studies where you could potentially even have just a placebo effect. So to take the NETTER-1 study, we looked at various quality of life metrics. And one way of looking at this is comparing time to decline in quality of life, because despite treatment, there is, in most cases, quality of life continues to worsen, although with treatment, it can be slower than without treatment or with a conventional treatment. So in the NETTER-1 study, lutetium dotatate was associated with a delay in decline in health, global health, meaning quality of life, physical functioning, meaning ability to perform normal daily activities, role functioning, meaning ability to perform hobbies such as, cooking or things like that. And we rely also on patient-reported outcomes such as patient diaries to look at symptoms like abdominal pain, diarrhea, and flushing. And you can see in these cases, abdominal pain and diarrhea, there was a clear benefit with lutetium dotatate versus high-dose octreotide, less so with flushing. These quality of life questionnaires often ask patients to fill out surveys with multiple questions. The standard EORTC cancer questionnaire has 30 questions. Then there's another 21 that are supposedly looking at more NET-specific questions. That comes up to 51 questions. That might be a lot, and not all of them are relevant to NETs. So for example, constipation is not particularly relevant. When we looked at the NETTER-1 study, we ended up looking mostly at a small number of symptoms, such as flushing, diarrhea, abdominal pain, bone pain, fatigue, that we felt were relevant to patients with midgut neuroendocrine tumors and not using a lot of the questions. So patient input in trial design is increasingly utilized, but I just want to point out some conflicts between what might benefit the individual patient who's enrolling in a trial versus, let's say, the data or the ability to come up with useful data for the general population. So many patients, for example, understandably don't like placebo-controlled studies because there's a chance of being randomized, obviously, to not getting an active drug. That's not great for the patient that's enrolling in the study, but at the same time, it's often the best way of evaluating a new drug and getting it approved quickly. So the most convenient way to look at a new drug is to compare it in patients who have exhausted all standard treatments to no treatment at all. That obviously results in the highest chance of the drug showing benefit and eventually being approved for the general population. When you're comparing two active drugs, it takes often a lot more patients being followed over much more time with a lower chance of the drug being shown to be active. So when you ask, why do we have placebo-controlled studies? That's the reason. Crossover is another issue. When the endpoint is progression-free survival, I personally think that crossover should almost always be part of the trial. In other words, patients randomized to placebo, if they progress, they should be allowed to cross over and receive the active drug. Regulatory authorities often don't like that, and they sometimes mandate non-crossover because they want to see what impact the drug has on overall survival. So there's a tension there, again, between what's best for the patient enrolling on the trial versus what the regulatory authorities want to see and what's best for generating a clean trial that results in drug approval. Another issue is eligibility criteria. And this is a big peeve of mine because we have a lot of trials with a lot of eligibility criteria. And sometimes I feel like there's a lot of copying and pasting going on from other trials with things that are not necessarily relevant. And when you think about it, it's like running a hurdle. You know, you have to be able to cross over sometimes 20, 30 eligibility criteria. You can end up not being eligible, not for a reason that's actually relevant to the trial being a success or the patient, you know, being safely enrolled in the trial, but that's something that's completely irrelevant and nonsensical. So to take an example, there are some trials that have nothing to do with blood pressure that unfortunately require that patients have a perfect blood pressure to enroll. Who knows why it was inserted there? Sometimes it was just perhaps copied and pasted from something else. So these are sometimes reasons why patients may end up not screening for trials. It hurts everyone. It hurts the trial, hurts the patient who really needs to be on a trial. So we need to be very careful about what our eligibility criteria are, and maybe patient input can be helpful to hold investigators and sponsoring companies. And there's one more thing I want to point out regarding eligibility criteria. There's often a requirement that the patient not only be eligible at screening, but also eligible on day one when they enroll in the trial. So just to give an example, this morning I'm dealing with a case of a patient who's screened for a trial, found to be eligible, and today one of the labs is just slightly off. And now we're scrambling, trying to repeat the lab. I really hope she's eligible, but it would be a really tragedy if she's ends up not being able to enroll in the trial for a very trivial lab abnormality. So we really need to think about all these things and as companies to think about what's really right for the trial and for the patient when it comes to these eligibility criteria. There's also many other things that we need to consider. Increasingly, we've allowed patients to be monitored on trials on televisits. I think that's a fantastic thing. Many trials require that patients come to the center or be evaluated very frequently. Clearly, patients don't need to be seen in person for each and every one of these visits. So trials can be extremely burdensome. Televisits are one way of alleviating that burden. And I really hope that this continues. There have been efforts to reverse this now that the pandemic is abating a little bit. I really hope that we get to continue monitoring patients on Zoom and other televisit modalities. Also, we really need to consider the number of labs, EKGs, monitors, PK testings, safety visits. A lot of this stuff is above and beyond what's required, I think, for patient safety and trial data. So I think we need to look carefully at what we're demanding of patients, and I think patients groups can also look into these trials and provide input and ask, what's really necessary and what's extraneous. What about areas of need? We clearly need studies for poorly differentiated neuroendocrine carcinomas. That's an area where we haven't really made much progress in the last 3 or 4 decades. I'm happy to say that we're increasingly seeing studies in this area, but we really need more. I'd like to see more cell therapy studies, things like CAR T studies, bispecific antibodies, T-cell engagers. We see a lot of studies with very similar tyrosine kinase inhibitors, things that are not that novel, and we really need novel drugs. And cell therapies are really one area where there's the potential for long-term remission, not just controlling disease for a short period of time. It's a long shot, obviously, when we're talking about completely new therapies. But we're working preclinically on developing CAR T cells that are targeting the somatostatin receptor, as well as bispecific antibodies, and others are working on that as well. And hopefully, we're going to see some clinical trials in the near future.